Good evening, everyone. And on behalf of CPOEM Foundation, which stands for Creative Portal for Oncology Education and more, we invite you to this webinar on unraveling the genomics in lung cancer and liquid biopsy in non-small cell lung cancer. So we have two very eminent speakers. We have Dr. Ramaswamy Govindan from Washington University School of Medicine. And we also have Dr. Ullas Batra, who is the Chief of Thoracic Medical Oncology at Rajiv Gandhi Center in Delhi. So our educational grant for today is by Garden 360. The first speaker for today is Dr. Ramaswamy Govindan, who is the chair of medical oncology at the Washington University School of Medicine at St. Louis, Missouri. After his initial training in Jipmer, Pondicherry, India, Dr. Govindan pursued his internal medicine residency and fellowship in hematology and oncology at premier centers in the United States. He has received several prestigious awards and is a member of several national and international committees. He has authored several textbooks and is also the editor of the Washington Manual of Oncology. His area of interest includes proteogenomics in lung cancer and currently working on the Cancer Genomic Atlas, the TCGA, as a co-chair sponsored by the National Cancer Institute. He's working on sequencing the whole genomes and exomes of patients with lung cancer. And moreover, his approachability and humility magnifies his greatness. So, and we also have Dr. Ullas Batra, who is trained from Kidwai Institute of Oncology. And he has done several preceptorships in various cancer centers like the MD Anderson Cancer Center and in the Royal Adelaide Hospital in Australia. He has participated as a principal investigator and co-investigator in various phase 2 and phase 3 clinical trials. He has numerous publications to his credit, and his area of interest is mainly in lung cancer. And he is actively has participated in organizing various seminars, CMEs, conferences throughout India. He's also a thesis guide to DNB residents. He's a very good orator, and he's also quite witty and very humorous. So without further ado, I would request Dr. Ramaswamy Govindan to give his lecture.
Thank you. Thank you so much for your kind introduction. Great to see all of you. Great to see Ullas, who appears to get younger every year, and it's always good to see you. First of all, glad to be with you, and I'm glad that we have the opportunity to discuss this. I've given several lectures on the topics of genomics of lung cancer to this audience, a similar audience in India for a number of years. So today I thought I'll just take a slightly different tack and explore what the proteomics of cancers can do on top of genomic analysis. And as Vishwanath said earlier on, we've been working on the genomics of lung cancer for the last 10 years, and the Cancer Genome Atlas project has published a number of papers, including 4 seminal papers on the genomics of lung cancer. As you know, the genomics of lung cancer, technically speaking, it looks at the genes, which is essentially the DNA, either the whole exome or whole exome or the whole genomes. The RNA is a transcriptome, and now we are moving on to the protein. As you know, DNA codes for RNA, and RNA is translated into proteins. And the proteins are the business end of these molecules, and it is the proteins that drive all of them. So I'm going to show you a few examples of how much our understanding has changed. So this is not for practice application. I just want to caution you, this is more for you to understand where the science is, and more to understand what is happening now in the research field at the cutting edge, and where we are going to see a number of these research initiatives that are going to go on in the coming years. I have no conflicts here. And I would go back and show this slide once again, that the Cancer Genome Atlas project was a massive enterprise studying 10 different cancers, 10 rare cancers and 33 different tumor types, as this enormous collection of data from 11,000 patients, and have several thousands of papers coming out of this effort from variety of cancers, including lung cancers, and 2 types of lung cancers with a focus of TCGA, the adenocarcinoma and the squamous cell cancer. And the small cell lung cancer was not studied. And some of the work was done by our group, as well as the group from Germany and the group from the Memorial Sloan Kettering. And so those are the seminal papers in small cell lung cancer. So today I'm just going to add the layer of proteomics into genomics. And, I just would highlight a few examples. I'm just going to give you a few examples here and there. And in the past, and even now, many times clinicians look at genomics as a means to find targets for therapy. And that's certainly an important effort. But I just want to tell you that by doing this comprehensive analysis of several thousands of whole genomes and several hundreds of thousands of whole exomes and partial exomes, we have now come to one realization that we are not going to find readily actionable targets in lung adenocarcinoma or squamous cell lung cancer at the DNA level. And I want you to understand what I said or think about this carefully. At the DNA level, we may be able to understand the biology better by studying the RNAs and the proteins, and we may find novel targets that way. But looking at the whole exomes and whole transcriptomes, I think for large part, we'll be reconfirming what we have already identified. And this is an important initiative in practice. We are going to be able to find identifiable targets for therapy. So that's still very, very important, either through the tumor genomic effort or through the cell-free DNA. Ullas will be talking about that. But in terms of research, we may find additional areas of this, and I'll show you a couple of examples in a few minutes where we'll find targets or the key drivers for oncogenesis. We will understand a lot better the immune microenvironment by throwing the RNA sequencing and proteomic analysis, and I'll give you a few examples, and that would help us set the stage for novel therapies for immune targeting and beyond the PD-1, CTLA-4 axis, and we'll be able to customize therapies as we move along in the next decade. The third, I think, a very important area of my own research is to understand the biology of metastasis. After all, patients with cancer die often from metastatic process, and that's something we have not fully understood, the processes governing metastasis and how we can approach that. And I think by studying the proteogenomic alterations, we'll be able to look at that more concretely. And we have actually, this is one of, again, my areas of research, where we study how the tumors metastasize to the lung, from the lung to the brain in particular. And we also studying comprehensively other areas of metastasis. So this would require a comprehensive analysis beyond the DNA analysis, beyond the whole exome and transcriptome. And that would require not only the transcriptome analysis, proteomic analysis, and also methylome analysis, which I'll show you an example. And then finally, we have not fully conquered or understood the mechanisms of resistance to therapy, particularly in areas of targeted therapies, where we have a better understanding of how to initiate the target therapy, but we don't have a very good understanding of how to go after the mechanism of resistance, and that also would require comprehensive approaches. For those who are fairly new to the field of lung cancer, and I just want to highlight this using this study from the TCGA that we published almost 6 years ago. Lung adenocarcinoma, if you really want to define this, is a disease of the receptor tyrosine kinase and RAS pathway. And on the left side, you see various members of this genes, and you can see on the right, on the horizontal axis, is the patients. And you can see that these are each square is a patient sample. And if you see that if you have KRAS mutation, you don't have EGFR mutation in this population. So it's mutually non-exclusive, mutually exclusive. And these alterations are not just point mutations, as you see here, but they can involve fusions, as many of you know, amplifications, particularly in genes like FGFR, MAP1, and PDGFRA, and some tumor suppressor loss that indirectly activate the RAS pathway, particularly the NF1, RASA1. And then they're often sometimes accompanied by STK11, particularly the KRAS mutations, as you can see here. So, but for large part, it is a receptor tyrosine kinase pathway, RAS pathway alterations. And this is the work out of 1,000 samples. We did find a small number of those samples that we didn't find a targetable alteration. So that was a subject of a paper that we published earlier last year. And we looked at a small number of groups of patients where we didn't find the targetable alterations, and we subjected them to whole genome sequencing. And we found that additional members of the receptor tyrosine kinase pathway, RAS pathway alterations in a good number of those samples. And then we left with a truly RTK RAS negative adenocarcinomas, and we studied them in great detail. So there is a small group that does not have known members of this activation through the analysis we performed so far. We were able to retrieve some examples, including some instances where we missed KRAS G12C in the previous TCGA because of the low coverage in the whole exome sequencing of the technology 10 years ago. Now you can see that the coverage is very good with the whole genome. So we picked up a number of those samples that are potentially targetable. But then instances where we did find where we had not appreciated earlier on is those examples where the RTK pathway was activated. Here's an extreme example of a patient sample where the EGFR amplification was far end, really off the chart, whereas the bulk of the rest of the sample was here. The amplifications are significantly elevated or amplified, and you can see the copy numbers are really off the chart compared to the rest of the samples and accompanied by these alterations. And we also saw in one example a deletion in a gene called RASA1, which is significantly low expression, and that also activated the RTK pathway. So there are a number of different ways this pathway can get activated beyond the mutations. And then there are some samples that did not have any activation of the RTK pathway. And one example is, I won't go through how we arrived at this. It's a very complicated process. Basically, we are looking at this open chromatin regions of various genes, and we found that there was a gene called ILF2, which is involved in DNA repair. The promoter region, that's the earlier part of the gene sequence, where there were some alterations that we found in 6 samples in this, and that was associated with corresponding alterations in the expression. And this is an important pathway for DNA repair. And this is one example how we are able to find some unique alterations that we did not appreciate before. Is it something actionable now? Not really, but it is something, that unique alteration that was seen in 6 samples in this that did not have any known members of the RTK-RAS pathway alteration. The other ways these cancers can evolve is sometimes they can activate the oncogenes. Like, for example, here you can see a MYC oncogene activation in those so-called oncogene negative group, the RTK-RAS pathway negative group. And we also saw a number of tumor suppressor loss here in various samples, including P53, KEAP1, STK11. So I suppose in some tumors, you don't need an oncogen activation. You can just have a tumor suppressor loss itself, especially if a couple of them happen at the same time, that could drive the cancers. But they are very complex. The story is very complex. Once again, these are individual samples on the horizontal axis. You can see a given sample here as loss of P53, KEAP1, STK11, and activation of certain oncogenes. And it's a combination of those things that lead to creation of this cancer, or transformation, I should say. And where no matter how hard you look, you may not find these alterations. Or perhaps with better technology, we may find some alterations in that using proteomics and other approaches. So that takes us into the second part of our talk, where the first part, I just want to show you that with better genomic technologies, we may be able to retrieve some examples of how these cancer cells, or what drives these cancer cells. But as we move along, now we are able to do, using mass spectrometry, we can look at the proteins. And it was possible earlier on, but we were able to do only a small number at the same time. But if you look at this, I would refer you to these 2 papers that we published in 2020 and 2021 as a part of the national effort, along with a group from Asia. Then we co-published the paper in Cell back in 2020. And in this effort, we looked at 10,000 proteins. Remember, we all know that there are 20,000 genes in each and every cell. And these DNA, 20,000 genes, they create multiple transcripts, and they create multiple proteins. At the moment, we don't know what the total proteome in the human cell is. That has not been estimated, but it is obviously going to be multiples of 20,000s, because genes can encode different types of proteins and different fragments. And we looked at this as 10,000. It's still a drop in the ocean, to be honest with you, but I think it's still a massive effort compared to what was possible in the past. More importantly, proteins don't necessarily, when they get activated, they get phosphorylated or acetylated, or sometimes in the process of destruction, they get ubiquitinated. And now we have technologies to look at this phosphorylation, acetylation, all of them. So we looked at 41,000 phosphorylation event, 11,000 proteins in total, and all these samples underwent whole exome, whole genome. That is a beginning point, and then we added multiple different things. So in a given sample, this is a graph that tells you in a given sample you had whole exome, whole genome, transcriptome, microRNA profile, and various different things, including the proteins, phosphorylation, acetylation, etc. And so this is a massive effort, and I won't go through this in great detail, but the point of that is to show that such a comprehensive effort is possible. And what have we learned? I'll give you a couple of examples. I won't walk through every single panel here in the interest of time. One important thing for you to appreciate here is this. For example, when we see a lot of these copy number alterations, you get the report from various different vendors and copy number altered. And again, not all of them may be meaningful. Here is an example of this. And if there are significant, if you look at this, and for example, if there are 6,000 events, and we'll take the cancer-associated genes, for example, 593 genes that have copy number amplification. And if you look at this, of the 593 cancer-associated genes we looked at, 457 cancer-associated genes, these are genes known to cause cancer, they have copy number alterations. But how many of them have associated RNA expression? About half. And how many of those genes that have copy number alterations and the RNA expression have the protein elevated? That's about half of that. So that comes to about nearly 1/4 of what copy number alterations. How many of them are really activated at the phosphoprotein level? It's fraction, 12 of them. So a lot of these copy number alterations don't necessarily get transcribed into RNA. Very few get translated into proteins, and very few have this post-translational modifications. So perhaps these 12 are the ones that matter. The 12 copy number alterations that carried over all the way to the phosphoprotein, this is the list here. Those are the ones that really mattered. And this is the way you can narrow it down for functional studies. Remember, these proteomic studies, the genomic studies are important when we know with the known associated genes. It's great, but then when we don't know the biology of the genes, that really gives us a short list of the genes that we can do functional studies. We can delete the gene, or we can decrease the protein expression to see what happens to the cell. And that's how we can minimize, or we can narrow down the things that require functional studies, both in vivo and in vitro, or in vitro and in vivo. And so this is an important slide. If there is one takeaway point from this slide I'm going to focus on, is this slide, that not all copy number alterations are associated with alterations at the protein level or phosphoprotein. It's only a fraction of them. So that's important. So not everything you see in the report is meaningful. So it's very important nowadays when we have these reports that really describe various gene alterations, going and targeting them all. It's not the right thing unless it has a biology behind this. So if there is one important message from this talk, it is this, that not every alteration is meaningful unless it's validated. For example, if you see EGFR exon 19 deletion, there's no question. It's a strong biology behind this. ALK known fusion, no question. But then if you suddenly see a gene that seems like it is important, unless it's a biology in that cancer, it's important for you not to really jump to conclusions. And remember this slide, this is an important piece of that. And the other important point is that there's a gene called PTPN11, which is a gene that codes for a protein called SHP2. And the SHP2, the PTPN11 gene is never mutated, and almost never mutated in lung cancer, less than 1%. It is mutated more often at a slightly different location, leukemia. But what we observed was quite remarkable. In a group of EGFR mutant patients, that's a group of patients, the EGFR mutant here, we saw significant phosphorylation at the Y62, it's the tyrosine at the position 62, phosphorylation, which is the red mark that you can see here, exclusively in EGFR mutant patients. You don't see that in EGFR wild type except a few samples. And if you look at the EGFR mutant samples compared to the wild type, you can see that it's really, again, off the chart. The PTPN11, Y62, there's a SHP2. This is SHP2 is an important protein in the receptor tyrosine kinase pathway. And SHP2 inhibitors, we and others are studying them. And so this gives us credence to combining EGFR inhibitors and SHP2. In fact, I have patients who are progressed on osimertinib and now on osimertinib with SHP2 doing very well. And these trials are currently ongoing. And this is a very important target. And we found that remarkably exclusive to the EGFR mutant population. We did find some in other cancers, but not as much as the EGFR mutants. And there's a lot of interest in studying, even though we didn't see this level of phosphorylation, there's interest in studying in KRAS mutant population as well. In KRAS mutant population, there is a gene called SOS1 that's also off the chart here. You may be able to appreciate in this graph. And the SOS1 inhibitors are major interest as well right now. And so this is an example where without the proteomic analysis, you wouldn't have seen this. Gene sequencing would not have told you that PTP11 is an important gene. And I think that's important to keep in mind. What about squamous cell lung cancer? It's a little bit of a tough case. And unlike the adenocarcinoma, where we can see a number of these alterations that are seen, squamous cell cancer is marked by alterations predominantly in these tumor suppressor genes, or genes that are not easy to go after, like the NFE2, L2. And there are some alterations that are shared between the two, but it is in general not that easy to tolerate. I would refer you to this paper if you really want to go back and read, and I think there was an editorial, if I'm not mistaken, and you can read this. And one important point I want to highlight is that when we did the genomic analysis in the earlier TCGA paper, with the technology available at the time, we saw a peak in the region, which is very close to FGFR1, and there was a lot of interest in looking at the FGFR in this population, but the FGFR inhibitors have not really turned out to be that effective in lung squamous cell cancer. And the question has always been, is it because of the drugs, or is it because the target is not really FGFR1? Remember, I showed you in the previous thing, not every amplification results in meaningful protein activation. I showed you the previous slide that less than, a fraction of them from 400 plus to only 10 of them or 12 of them translated into proteomic or phosphoproteomic analysis. So we did the same thing in the lung squamous sample, and we asked the FGFR amplified tumors, or the region where it is amplified, what happens at the protein level? It turns out the FGFR is right here, and not that much overexpressed, but one gene, NST3, which is really again off the chart. And we think that the amplicon here, which really shows a high FGFR, is associated with NST3, and that probably is the major area of this oncogene driver here. And so there's a lot of interest in looking at NST3. So by genomic analysis, you may be inclined to think it is FGFR1, but in reality, at the protein level, the clarification comes, it is probably the NST3. So there are studies that are currently ongoing, going after that in a great extent. My apologies, I'm recovering from a bad cold. And so here is that other thing, we can really look at this various different alterations in one go. CDKN2A is altered in squamous cell lung cancer. So there is a lot of interest in studying CDK4/6 inhibitors in tumors that have lost CDKN2A tumor suppressor. As you know, it comes in two different forms, CDKN2A, the P16 and the P14. And there have been a lot of studies looking at the various different isoforms here. But the part of the slide, you don't have to remember the details here, but you just want to appreciate what you can study. Again, the bars, horizontals are individual tumor samples. And you can see this, RNA expression at this level here of this protein, the P16 form, RNA expression, P14 form. The height of the bar indicates RNA expression here. And the color codes here, which may be harder to see, the blank ones are white and the red ones are amplified. And you can see that mutations are colored in here in yellow, and the green is a different kind of mutation. And the promoter methylations are here in purple. And you can see that when there's a promoter methylation, the RNA expression is blunted, the protein levels are almost absent. These are the protein levels you see here. So this is a very neat biology. So we have promoter methylation, RNA expression is very limited, proteins are almost non-existent. Wild type, we have a high level of RNA expression, the protein levels are really, off the wall here. And the same with P14 as well. So you can really see that how beautiful the technology is, how good the assays are. It reflects the biology that we see. But in the same samples, we have a wild type expression here, and we have proteins that are nicely present, both P16, P14. And you can see that, if you look at this RB, which is an important break here, in some instances, RB is gone. And this is the RB protein level, you can see significantly lowered. And in some instances, where you see that there is a promoter methylation, decreased expression, you can see the RB is lost in some of those things or poorly expressed. And in those tumors, you won't be really able to see much activity with CDK4/6 inhibitors. And this really gives us a nice characterization of not only the individual accompanying proteins that are called cis alterations and other protein alterations, the trans alterations in various genes, and that may be able to parse out in a future world. We'll be able to do this multiple assays to really say that these are the different drugs that can potentially be active, of course, after all the validation studies. And one of the areas that we are very interested, and I'm mindful of the time, Vishwanath, I'll finish up in a couple of 4 minutes or so. So here you can see that SOX2 is a transcription factor that is overexpressed or amplified in a number of these tumors. SOX2 is a transcription factor. It's very hard to go after transcription factors. So one of the things that we are very interested in seeing is that, are there other alterations that are present that really we can go after in these SOX2 altered tumors? And one of them is the EZS2, which is again a number of targets, a number of drugs are being targeted here. And, this is an important area of research that can be used, EZS2 targeted therapies in SOX2 amplified tumors. And then very briefly, when we looked at the immune landscape, we have the tumor tissue here, and we have the normal tissue here. And when we regrouped them, and there were 3 types of, subgroups we saw in the immune cluster. One group within the tumor, we have a hot tumor, we have a cold tumor here, and this is a normal tumor tissue. And if you can carefully pay attention to that, again, the greens are, the blue ones are where things are lower expression, and red is overexpression of these corresponding members here that are highlighted here. The hot cluster has expression of CD4, CD8, dendritic cell. The cold tumors don't have any of those activations. But if you look at the normal adjacent tissue, they actually have somewhere between this hot and the cold extreme. And this really suggests the possibility that in certain cold tumors, there is the immune downregulation. It's not like immune system is not activated. There's a suppression of the immune cells here to enable the tumor cells to grow. And we are very interested in this cold tumors to see what's happening. And then we go further down in the metabolic pathways. We see certain pathways in the metabolic signaling pathway, including the lipid biosynthesis, PPAR signaling, and something to do with the mucin glycan biosynthesis. They seem to be more active in the group that is cold tumors, and they in turn very likely suppress the activation of the immune activation. One of the things I want to caution you is that correlation is not causation. So these studies are predominantly correlation studies. That does not mean that activation of PPAR signaling causes downregulation. The findings of the associations are intriguing. That gives us opportunities for us to study in the lab, because studying one signaling sometimes take 5-10 years and a number of people in the lab. So this really opens up possibilities. Somebody working in the PPAR signaling pathway looking at this would want to focus on some elements that may be of interest, and that's how the science advances. That's why when we publish these papers, we make it open access, we make all the data available to the investigators so they can really study them quite a bit. This is an idea to enforce or encourage the community to do lots of research studies, and that's the more important point of these slides. But this is very intriguing that a number of these pathways are overactive in the cold tumors, and that's an important thing to appreciate. Drilling this down further, we can look at this, what happens in STK11 tumors. As you know, if you have STK11 alteration, the chance of response to immune therapies are lower. Again, we don't withhold immune therapy from this population today, but there is increasing evidence that this is a group that may not do well on immune checkpoint blockade currently we use in lung cancer. Why is that? And again, you can see that these are the tumors that have a lower expression of CD8, macrophages, NKT cells, as well as dendritic cells compared to the tumors that are STK11 wild type. And we further showed that in that population with KRAS, with mutant, for example, this is the KRAS, all are KRAS, here's the KRAS mutant with STK wild type, and you can see that the immune score is much higher compared to the KRAS mutant and the STK11, KRAS mutation, STK11 wild type, the immune score is high. KRAS mutation, STK11 mutation, the immune score is much lower. You can see that. And whenever the STK11 mutation is there, the immune score is quite lower, and that may be related to activation of the neutrophil degranulation pathway, and that may create some unfavorable milieu for the T cells to be there. We are also entering the area of immune checkpoint blockade. beyond the immune checkpoint blockade, we are trying the cellular therapies using various different products, including BiTEs, CAR T therapies, as well as engineered T cells. And there are a number of targets that we find that potentially could be used for adaptive immunotherapy. And that's another important service the proteomic studies do. And I showed you the cold tumors, and we are very interested in studying why are the tumors cold. And I showed you some pathways that are activated, metabolic pathways. And we also drilled down further into what other activations. And it turns out that the cold tumors have a high Wnt signaling pathway activation, and that's what's shown on the other side as well. And there is a lot of interest in studying that in both lung cancer and non-small cell lung cancer. And this is a paper we published a few weeks ago, looking at what happens when the lung tumor cells go to the brain. And I'll give you the punchline here. It turns out that these tumors, when they go to the lung, they often have this primitive stem cell-like appearance, and they have a lot of open chromatin, and they go to the brain region, they respond to the brain ligands, then response to glia and other microenvironment, and they epigenetically alter some of the developmental genes important for the neural growth, PAC6, for example, and that in turn results in compared to the normal and the primary tumor, the significant epigenetic alteration of the brain metastasis here. You can see in the PAX6 gene that are important for developmental in the in the brain. So essentially, these are the cells that are primitive with open chromatin. They go to the right place, they get the right ligand, and then they try to activate and behave more like the cells that are native in that area. And that really is a neat biology that we unearthed recently. And in fact, we are now launching onto the fourth version of the CPTAC, which is similar to TCGA, but really goes on to proteomics. And in the recent CPTAC, we and a few others got funded to do analysis of samples from clinical trials, including the Alchemist samples. We'll be looking at various different markers and to predict outcomes in these patients. Briefly, in small cell lung cancer, relapsed small cell lung cancer is very hard to treat. We don't understand the biology. We published the very first paper in this field. Looking at this at relapse time, we saw the downregulation of the APC tumor suppressor, activation of the Wnt signaling pathway that confers resistance to chemotherapy as well as immune therapy. And in a single patient, now we do single cell sequencing where we look at a few thousand cells. And this is a single patient who did not respond to any therapy. And ASCL1 is a transcription factor present in sensitive tumor cells. And in this patient, very few cells have ASCL1. NeuroD1 is present more in resistant tumor cells. You can see in this patient, large numbers of the cells have NeuroD1. In addition, they had activation of the mesenchymal signaling pathway, Wnt signaling pathway, EZH2 activation, and various members that contribute to resistance, including MYC amplification. So this shows once again, in a given patient who did not respond to therapy, multiple mechanisms of resistance coexist. And this is no surprise that this patient did not respond to therapy because not one, 5 different mechanisms of resistance are in play in this patient. And so I'll close here just to show that. I hope I showed you a few examples how we can find more targets by doing genomic analysis. We can also by studying proteomic alterations, we can clarify which one of those genomic alterations are noise, which one is signal, and we can look at the phosphoproteomic alterations to study the tumor landscape significantly to find mechanisms that contribute to resistance, what we can do to activate or add to targeted therapies. And I showed you a few examples of how immune landscape can be parsed very carefully to understand the biology. And I gave you a few examples of resistance and an example of brain metastasis from lung cancer. There are several such examples. This field is rapidly growing. The next 10 years, you'll see a number of these papers looking at this in various different settings that help us inform biology. Of course, these efforts are not possible with one person. I lead a team of several hundred people at various levels, both nationally and globally, as well as regionally. And these are people who have contributed significantly to all my work and did a major part of the work from Washington University, NYU, MD Anderson, and Broad, MIT, Harvard Medical Center, and the NCI. A lot of our funding is supported by National Cancer Institute and the grateful patients and families. I'll stop here. Thank you so much.
Thank you, Dr. Govindan, for that insightful talk. There is so much being done in this exciting field of lung cancer. There are a couple of questions in the chat box which we can take if time permits. Can I go ahead with the questions, Dr. Govindan?
Yeah, I can see that. You want me to answer it?
Yeah.
So the first, I think Ullas has a question. Maybe I should answer Ullas first.
Okay.
Before I go to the chat.
Thanks, Dr. Govindan. And it looked like from a different world altogether. I actually have goosebumps, and I must place on record. When you came for the Rajiv Gandhi conference and you did the oration, that was a changing, defining moment in my journey in lung cancer, prompted me to do a lot of research. And your today's talk has again reiterated that there is a lot to be done. Fantastic. That was a comment. And a question now. That when do you think proteomics is going to be coming in of age in the clinical practice? Right now the point is to do genomics, NGS and everything, but do you think proteomics will come into the clinical field?
Yeah, absolutely. Remember when I always give an example. In 2008 we published the first paper in Nature, single example, one patient, whole genome. It took us 1.5 years and $1.5 million to do that one sample. Now we don't do whole genome in clinic typically, but we do the sequencing so commonly everywhere around the world increasingly. So it happened within a matter of literally 10 years. So it's going to happen. The question is really, how do we use it? Where is the need for it? Remember, the technology for the sake of technology is a pointless exercise, and technology in the service of a question or answering a problem is the right thing to do. So as we parse these things out, we may end up doing it, and it'll be context dependent. We don't need mass spec analysis for every patient. We may end up doing, for example, if you're looking for, let's say, SHP2 inhibitors come into existence, and we find that SHP2 expression is an important thing. Sorry. Sorry. So we may end up doing a SHP2 either by some kind of IHC or some kind of analysis. And proteomics is no new field. Well, proteomics is a new field. Protein analysis is not new because we've been doing IHCs for a long time. So mass proteomics will come perhaps at some point, but I think we need to know exactly where to use that first.
So let's say, Dr. Govindan, if you have a patient whose ALK for IHC was negative, but the NGS picked it up, what would your interpretation be in this patient? Should we do something more? Because obviously the protein has not been formed. And let's say that the test was done twice in a good lab, and we have taken out all those errors.
Yeah, I'm glad you asked the question. Ullas, by the way, please don't be formal. Call me Govindan. I call you.
You've been asking me for last 3 years, Dr. Govindan.
No, you should. No, please. So anyway, the point is, I think I want to be very careful. I'm so glad you brought this up, because what I told you is that I'm talking about this proteins that have genes that have not been validated, where we really see copy number alterations. You shouldn't get really excited about it. May or may not play a role in this. But things like ALK, there is also false negative tests in IHCs sometimes. And so if you have one or the other, I mean, if you have particularly the nice, reliable lab, if you have a fusion transcript, and absolutely I'll go for it. And the proteins, the IHCs may not necessarily be properly done, or the issues. And if there is a fusion transcript, I absolutely would act on it. I wouldn't wait for the protein level for this, partly because of technological issues. I'm not speaking from both corners of my mouth. I'm talking about the mass proteomics, which we have. This is, I can go on for an hour and a half on the technology we used, the validation that we used, the quality control we used for these kind of this is extremely rigorous. That's why you see all these things, and that doesn't happen in a routine clinical setting. So again, this is immunohistochemistry at a low resolution level. So without a question, I would go by the sequencer in those instances, the known biology and known gene.
You can answer the questions now, Dr. Govindan.
Okay, very good. So the first chat question is that, do you think copy number alterations identified in the DNA NGS should be verified by other methods? If so, what would be the one? I honestly would think, to be honest with you, when you see these reports, stick to the basics. When you see EGFR mutation, go after it. ALK, go after it. ROS, RET, etc. KRAS, G12C, if you have access to sotorasib. Don't get distracted because you suddenly see a gene that, let's say, for example, LKB1, don't start adding drugs on a women fancy. You should be very careful about combining drugs unless they've been validated in a clinical trial. It is the right approach. It is the conservative approach. It is the right approach. This willy-nilly combination of a single patient is not a good idea unless it's partly carefully thought through. So copy number alterations in DNA, I wouldn't bother to verify it at the current times. And again, there is no proof that EGFR, I'll give you a simple example. EGFR mutation, EGFR amplification. I don't know what to do with that. When I see high level of EGFR amplification, I don't give them cetuximab or panitumumab or EGFR inhibitors, because we have not shown that that itself identifies a group of patients who benefit from that. We probably haven't done the right studies, but we don't do those kinds of things off the beaten path. What would be nice is that if there is a clinical trial that includes those patients, get them on the trials, but we shouldn't be acting on them. And I think it's important to keep that in mind. And, the other thing is that we are now seeing ADCs, antibody drug conjugates. And if there is an antibody drug conjugate against HER2 gene expression, and if you have, then you look at the correct markers for that particular approved test. And then if that is, then you go after that. So that's how I would use copy number alterations if they are potential thing. Or genes named after proteins are isolated. Now, how are all the names of genes called by non-geneticists? There is an international nomenclature for the genes and the associated proteins, so that's extensive. It's available through various resources online, and you can Google that and find that. Role of epigenomics in recurrent non-small tumors and non-small cell lung cancer. It is an area of study, and I think epigenetics are harder to study. It's not that easy, and you have to correlate with the expression and the protein level at the RNA and the protein levels as well. And these are all investigations for clinical research. And again, like all research, you have to have a central question, you have to have a good methodology, you have to have a very clear endpoints, and this is not for clinical use. Final question, HER2 amplified in NGS but not picked up by IHC, what would be the approach? And I think this is an interesting question. Are these really translated into this? What do you do with HER2 amplified tumors? Let's say we have bona fide HER2 amplified tumors. I honestly think there's only one known approach in my view in lung cancer. That is, there have been some reports of using ADCs, small phase 2 studies showing that the HER2 targeted ADCs are effective in lung cancer, just as effective in breast cancer. So I would use that if it is appropriate. Especially if they have. Oftentimes they go by the IHCs. If the IHC or FISH is negative, I would ignore that, and that's probably a noise and probably not real. And again, I would just make sure that we do the appropriate testing in the lab. But the pure amplification alone in lung cancer, breast cancer may be a different story. If FISH negative, IHC negative, I would not go after that. I don't want to cut in Ullas' time. Ullas.
Please take my time.
No, I'm looking forward to your talk.
Thank you, Dr. Govindan. It would be great if you are able to stay back with us.
I'll stay. I can stay. Absolutely. I want to listen. Yeah.
Right. I invite Dr. Ullas Batra to deliver his talk.
Thank you. And thank you for the kind invitation. And thank you predominantly because it has come after Dr. Govindan's talk. And I mean, I am zapped. I am actually zapped after listening to him, and I feel like such a novice speaking after him. I have goosebumps right now listening to him, and that's a compliment, Dr. Govindan, and you know it, I mean it. So liquid biopsy in non-small cell lung cancer, and mind it, my standard of talking will not be as great as what Dr. Govindan has said or what he has elucidated. Mine will be more practical applications of liquid biopsy in non-small cell lung cancer. And my first reactions when I heard about liquid biopsy in non-small cell lung cancer was like, wow, immediately, like almost like what Dr. Govindan actually spoke. And it was like, everybody was waiting for the liquid biopsies to come and take all the pains of tissue biopsy away. My subsequent reactions on liquid biopsy in non-small cell lung cancer is, okay, the theory is good, but show me the evidence. So let's talk about liquid biopsies. So we all know the first law of oncology is biopsy. If you don't do biopsy and if you do start a treatment on FNAC, probably you will flunk the exam. But we also know that the second law of oncology is that tissue is always an issue. And it is not only, the things are changing, but that's not only in India, that's all everywhere. And if we look at the data, this data was presented in the ASCO conference. 22% of patients in USA had insufficient tumors for genotyping. Even if you do a biopsy, there's an intra-tumor heterogeneity. You may not always hit the right spot, coupled with the fact that there is an inter-tumor heterogeneity. That is, the tumor within the lung and the tumor within the liver may not be of the same way. So that is why, is there another way to look at this? So these are the problems that you associate with tissue biopsy. And is there any other way to look at this? And definitely there is another way. There's always another way to look at it. And that basically is liquid biopsy. So liquid biopsy is a very nice, technologically sound, very cute sounding, minimally invasive, without any problem to the patient, high consistency, overcomes the tumor heterogeneity, faster turnaround time. ctDNA may reflect the tumor aggressiveness biology, and dynamic monitoring also is possible. So let's come down to the basics. What does liquid biopsy actually? It's very fashionable to say about liquid biopsy, but what does liquid biopsy actually analyze? Does it analyze the CTCs? Does it analyze the exosome, extracellular vesicles, or does it analyze the ctDNA? It actually analyzes everything. But for the purpose of this talk, we are going to talk about the ctDNA. We are not going to talk about the CTCs and the extracellular vesicles, and that is right now not really in the clinical phase. It is basically in the research phase, as Dr. Govindan would probably agree. So clinical utility of liquid biopsy in non-small cell lung cancer, you can have 4 utilities. First, can you do the molecular profiling? Can you do a biomarker selection and targeted therapy selection? Can you monitor for the early response? That is the dynamic monitoring. Third is, can you determine the early disease progression and resistance pathway? And the fourth is to determine MRD in early or advanced stage. So at detection, monitoring, relapse or progression, and in the advanced state, if you have an early to determine the minimal residual disease. Well, let's take the first one. Can the ctDNA predict a marker as good as a tissue biopsy? That is what we are saying. Will liquid biopsies replace tissue biopsy? So this was a complete study basically, which was a comparison of the MASH plasma versus the tissue study. And in this, it was basically found that there were 7,000 consecutive cases. I think this was by presented, it's a Canadian study, Garden, and which was basically presented 2-3 years back. And what it basically found out was that there was a positive predictive value for EGFR mutations or insertions or AL cross fusions was all in the range of 90 to 100%. So the issue with liquid biopsy is if it is very specific. If it is present, it is present. If it is negative, then you may go ahead and do a tissue biopsy. That's the basic bottom line about the liquid biopsy. And this is a complete study which basically evaluated the same. There was another study which was basically called the SLIP study. Again, liquid biopsy was almost equivalent to tissue biopsy. And the Garden 360, basically, there were the people who there were mutations. 25% of mutations were picked up by the tissue biopsy. 25.4% of the biopsies were picked up by liquid biopsy. And there was a high rate of concordance between the two. So liquid biopsy, by and large, picks up mutations almost similar to what the tissue biopsy does. The specificity is good, but sensitivity is only 80%, and that is what. So if you have a biopsy, if you have a EGFR detection on liquid biopsy, by and large, you can be absolutely okay in giving them an EGFR inhibitor. This was about the EGFR. This was a vision study in which the MET exon 14 mutations were detected either by liquid biopsy or by tissue biopsy. And what this study basically found out was that there was a similar overall response rate and median PFS when treated with tepotinib if your MET exon 14 was detected by tissue biopsy or by liquid biopsy. Again, to highlight the fact, if your liquid biopsy says that there is a mutation present, it is present. However, if it says it is not present, then we may go ahead and do a tissue biopsy on that. So that was the initial one about the biomarker detection and target therapy selection. The second, if you have given a biomarker, if you have detected a biomarker and you have given them an oral TKI, can this be used for a monitoring? And I'm very fond of calling it, a BCR abelification of NSCLC. In CML, we do BCR ABLs every 3 months, and we then change the response. Will a time come when we will be doing all this DNA monitoring? And if the patient does not respond, will we change the treatment? There's an Apple study which is suggesting that we might do that. But let's look at what does the dynamic monitoring of EGFR mutations or ctDNA monitoring shows in the terms of a EGFR mutant lung cancer. So this was basically the benefit study. Now what they are basically saying is you do the ctDNA clearance. That is, you take it at the baseline, do a ctDNA baseline at around 3 months. if your patient with the help of your drug clears the ctDNA at 3 months, those are the people who will do much better rather than the people who are going to have, who retain the mutation. Now, whether it is a complete clearance or whether it is a fall, the percentage fall, we don't know as of yet. But exactly if a patient clears the ctDNA mutation at around 2 to 3 months, those are the people who are actually going to do much better with your treatment. Similar study was there in similar results were basically got in the FLORA study. Again, the clearance of plasma EGFR mutation at week 3 or week 6, what they basically found out was, if you clear it at 3 weeks, it was PFS was 13.5 versus 9.5. If you cleared it basically at 6 weeks, 13.5 versus 8.2. People who cleared off the ctDNA mutations had a higher overall response rate and an improvement in the PFS and OS rather than people who did not clear it. Similar study was basically done in the Arlot and bevacizumab arm. What this study again found out was that if you have a negative ctDNA, these are the people who did the best. People who had a positive ctDNA cleared at 3 months did the second best. People who had the ctDNA positive and ctDNA positive at 3 months did the worst. So maybe at 3 months we have a if we do a ctDNA clearance, we can tell the patients that whether we should be continuing the therapy. Whether we change the therapy at 3 months, that is what the Apple study was about, and we will talk about that later. This was similar there in the ALK TKI treatment. Again, this basically found out that if you clear the ctDNA for ALK fusion also, then also you're likely to do much more better. Now, this is something which is very, very important. Now, liquid biopsy we spoke about in the blood. Now, let us say you have a patient who has a CNS mets, and if you have ctDNA at week 8 and you do not clear that, that basically means that these patients are going to do significantly worse than the patients who clear it. So the early clearance at 4 to 6 or 8 weeks, the time is not yet decided, will likely lead to a better prognosis. However, whether the change of the treatment at that time, if you don't clear, is going to result in a better treatment, that we do not know as yet. So we spoke about the biomarker detection, the complete study, the naive study, the SLIP study. We found out liquid biopsy, if it has got a high specific value. Now let's talk about, we also spoke about the dynamic monitoring. Now let's speak about the early disease progression and resistance mechanism. So this was the study that I was talking about. This was Apple trial, and this was basically presented around 2 weeks back in ESMO 2022. If you have a patient who is basically having a ctDNA positive, and on ctDNA the disease is still there, and you shift them to osimertinib, the PFS was slightly better. I have not yet gone through the entire paper. I don't think it's any published, but this was what the gist of the study was. Now, what about the resistance mechanisms to osimertinib? Now, Dr. Govindan spoke about the SHIP. We all know about the C797, G796, L8718Q. A lot of patients are also unwilling to get a tissue biopsy done. So if your patient progresses on osimertinib, and we know 40% of them will have a MET amplification, liquid biopsy can actually pick up all those things. And we have a lot of studies like COC study, the Savannah study, which will basically tell us the answer whether combining of osimertinib plus capmatinib or savolitinib is going to be useful in these patients. But liquid biopsy is easy, and it can determine the mechanisms of resistance to osimertinib also. So that brings me to my fourth question. What about the minimal residual disease or the molecular residual disease in advanced non-small cell lung cancer? We all know that the MRD is a prognostic biomarker in stage 1 to 3 non-small cell lung cancer. And this is what the GenSec basically showed. If you have done surgery and your GenSec is positive at 12 weeks post-surgery or at 6 weeks post-surgery, you are more likely to get a relapse of the disease. The PFS is going to be much more inferior. This is something which I'm very, very keen about. Now, let us say you have given a patient a TKIs, or, and this question is something which is going to be addressed for the FLORA trial also. If you have a patient who has taken osimertinib or gefitinib or afatinib for 3 years, and the PET scan is in CR, how do you, can you stop the treatment? So this was a study basically which said if your disease was in CR, who has been treated with frontline TKIs chemotherapy. There is no disease on the PET scan, MRD negative, CA negative. Give them a drug holiday, continue monitoring them for MRD. When they are positive, start with a drug treatment. If negative, again give them a treatment holiday, something like a BCR, ABR, what we say TRF, the treatment free time. And this is also something which is this was a very early study which was presented last year in 2021. There could be a median drug holiday of around 6.3 months. This study is not, as Dr. Govindan would agree with me, this is not yet mature for clinical use, but that's a very exciting patient. We all have patients. We don't know how to continue immunotherapy in second line, in first line till progression. The keynote trials basically showed that you can give it for 2 years and stop. The checkmate trials, on the other hand, in second line, tell them you can continue till progression. So if you have given treatment for somebody for 2 years, and then if you want to stop, I think MRD at that time will show a very good, should be a good tool in telling us whether we can stop treatment for someone. Given the cost of the treatment, I think that's going to be a very, very essential test. So again, the clinical utility of plasma ctDNA, you can stratify them according to the risk. You can use them for response assessment. You can use them for resistance monitoring. You can definitely use them for the MRD monitoring. Now that was all about the theory. Now we have done some work in liquid biopsies. Nothing as great as Dr. Govindan has done. Some small liquid biopsy we have done. We'll just like to show you that, share the data with you in another 5 minutes time. So the initially was a single gene testing where we got a concordance for the EGFR mutation detection, a single gene, not NGS, between tissue versus COBAS versus ddPCR. We had around 113 patients, and out of it 92 patients did a ddPCR, and 76 also underwent plasma genotyping by 76, single gene EGFR study. And what it basically found out was that there was a 95.9% sensitivity and 97.2% sensitivity of tissue genotyping versus COBAS. And now also, if tissue is an issue and if we want early reports, we do the RT-PCRs very, very commonly in our institutes, where we get the report next week. Today only we had a patient and we can start them on oral TKIs in ICUs. The tissue versus ddPCR, the sensitivity was 100%, specificity was less because ddPCR will not pick up other mutations actually, but this was also very, very important that we could use. This was an abstract which we presented for T790M by ddPCR. Again, the T790M detection rate in our patients was around 50%. This is not 60% as in the Western literature. This is not 40% as in the other literature. However, if you give osimertinib following plasma detection of T790M or by tissue detection of T790M, our median progression-free survival and overall survival were by and large equal. So there was no difference in T790M detected either by ddPCR or by tissue biopsy. Now, this is something which is very, very close to my heart. We have done dynamic monitoring for EGFR again by ddPCR and by ddPCR. This study, we had to interrupt it a bit early because of COVID. But that is what we had done. We had taken 47 patients whom we had done serial liquid biopsies for a single ddPCR, a single gene ddPCR. And what we basically found out were, again, which is there in the literature, that if you have an early clearance of ddPCR, you're more likely to do well. Also, if you have a slight increase in the ddPCR, in the EGFR percentage, you are more likely to progress in the next cycle. So we are coming out with a paper for this, hopefully in the next 2 to 3 months. This is also a study where we did a dynamic monitoring for nivolumab. Here we use a ctDNA and NGS. And this is again a study which is very, very close. We wanted to see after you give nivolumab in all the patients, and if you have after 4 cycles a stable disease or an IUPD, how do you really interpret? The salient features over here basically was that if you had a median ctDNA concentration which was higher, that correlated with a shorter PFS. If molecular response of 50% was observed in patients with PR, SD, and 2 patients of PD, patients who had a less than 50% molecular response had a median PFS of 3.9 months versus more than 50% response who had a median PFS of 5.5 months. Again, ctDNA levels at 4 weeks were predictive of progression in those who were showing IUPD at first evaluation and PD subsequently. Again, we are going to come up with a paper on this, and hopefully this This is something which is very, very, dynamic monitoring, which is very, very close to my heart, and hopefully we can come up with this. And we are taking a new study where we are incorporating this in all the cancers and not only in the lung cancer. So this was about our data. And to conclude, I think I'm too excited because of two things. After listening to Dr. Govindan's talk, and after listening, after doing work with liquid biopsies, and I'm sure in the next 5 years, the era will belong to proteomics, liquid biopsies, and lung cancer is going to be as happy. The patients are going to be as happy as this little girl is. Thank you. And with this, I hand it back to the organizers. Thank you.
Yeah. Thank you, Dr. Ullas, for that very nice and humorous talk, and very nice studies which you have elucidated. And definitely do agree with you that it is an era of liquid biopsy and hopefully proteomics as well. And it's not only lung cancer, like you mentioned, the punchline. We saw growing data in colorectal malignancy this ASCO. So I can see one question in the chat box. What is the minimal lower limit of detection needed for MRD? And can this droplet digital PCR, which is ddPCR platform, be used for that function?
So I am not a molecular pathologist, but ddPCR cannot be used for that. The MRD is not simpler as the ctDNA, cfDNA. We are not yet doing MRD, but I think Guardant and Thermo Fisher are coming up with the limits for that. So I really will not be. Maybe Dr. Govindan can take that question.
Dr. Govindan, would you like to take that question?
I'm sorry, I didn't know. What's the minimal LOD needed for? It depends on the assay. Sometimes as few molecules per ml you can detect, depending on how sensitive the PCR is. Maybe a few per, few molecules per cc, I guess. But I don't know the specifics of it, to be honest with you. But I think it depends on the assay, varies from assay. But I think, first of all, Ullas, it's hard to do these studies. So I congratulate you for doing this, particularly in the midst of heavy practice and all these challenges that you have. Here, we have a well-established system, so it's a little easier. Even us, we face a lot of challenges here too. But I think it's very hard to do anything there with such a very busy clinical practice. So it's really commendable you're doing this.
All inspired by that 1 hour talk, Dr. Govindan.
No. You're being too kind and too nice. No, not at all. But I think it's great. But I don't know whether it's I was going to keep my mouth shut, but I don't know whether I should say this or wait for the question. One thing I would caution you, I hope somebody would ask me the question. I thought I would respond to that, but I'm going to do it anyway. If you ask me, do I use MRD assays in the clinical practice? The answer is a big no. And I think it's important to appreciate one thing, that when you do a test, you should act on it. I think it's important. And we don't know fully how to interpret these tests. What we've seen so far is patients who have persistent high level of ctDNA don't do well. That's easy to say. In different contexts, right? For example, in a metastatic disease, was that helpful? Not really, because you're going to go by radiographic changes. Ask yourself how many times you see on the CT scan, you see some slight changes, you just ignore it. Patients are feeling well, you continue that, or you radiate. Number of times I've continued therapy for almost even a year or longer. And so in the absence, you're not going to change somebody's therapy just because the ctDNA level is going up. If it is, how high you should go up? Those questions are not answered. And more importantly, we don't have great drugs to follow up after that. What is the rush to give a second-line docetaxel, pemetrexed? So I think it's important to keep that in mind. So I would really ask you to think twice before embracing this in practice. Perhaps the reverse of what the conference is supposed to do. Liquid biopsy is very good in making initial diagnosis determination when you cannot get enough tissue for biopsy, molecular analysis. But you have to keep in mind the false negative rates are significant still, 20%, especially in thoracic only confined disease and lung cancer. So it is not the end all and be all. And you will see sometimes mutation like JAK2 that is not coming from lung, but coming from the heme malignancies. So these are CHIPs or the clonal hematopoiesis. So you got to keep this in mind. So serial monitoring, we don't do that in routine practice in any situation. We don't do this in stage 3, stage 4. It is still a matter of research, in my opinion.
Dr. Govindan, I have a question. So just extrapolating from colorectal malignancy, the recent data when they said at 4 weeks you do ctDNA, And that could be predictive of if it is positive, give chemo. If it is not positive in stage 3, you could probably avoid chemo. Do you think something like that would come up in stage 2 lung cancer post surgery?
The problem with stage 1 and 2 lung cancer is that the false negative rate, in other words, you have active tumor, but the ctDNA is negative, is very, very high. And unless the tumors are very large, even metastatic disease, predominantly thoracic only confined disease, the false negative rate, in other words, ctDNA not present, is pretty high. And so I would definitely be careful in interpreting that. So there is a large group of patients with stage 1 and 2 lung cancer who don't have ctDNA, even before operation. They have big tumors in the lungs, but you don't see. And you probably can see that the resolution gets higher and higher, and the sequencing depth is higher and higher. That becomes very expensive. And that's the reason you're not able to pick up many. In some instances, these tumor cells don't shed DNA, or they have very small amounts that are swept up by the normal DNA. So you got to be like every technology, you got to use it wisely. And it is a useful thing, a great tool, in my opinion, at the moment for initial diagnosis, and perhaps at some point for minimal disease assessment or stratification for somebody for intensification of therapy or de-intensification. We are not there yet. To me, the biggest potential, in my opinion, I see is in stage 3 lung cancer, where we want to optimize therapy. Persistent DNA at 4 months after chemoradiation, Adil Chowdhury from initial Stanford, now at WashU, showed in a nice paper that persistent ctDNA, again, these are small studies. Remember, not every study should result in change in practice. Phase 2 or early studies should not result in change in practice unless it's validated over and over again by large, adequately powered studies. And there should be a consequential treatment decision one can make based on that. In the absence of that, we should not just simply changing therapies willy-nilly for this. So stage 3, I think there's a lot of potential following chemoradiation. If there's a persistent DNA, then we may have to ask the question, should we escalate it to something else? And that again should be a part of a research study, not again in practice. I'm a little distressed that I see these kinds of one-off practice here and there all over the world based on one small phase 2 study or one presentation somewhere. You should not change this practice. And these are very expensive tests. And everything should be done in research context, study, validate large numbers. Practice changes should only happen after sufficient number of analysis or adequately powered analysis.
Sure.
Dr. Govindan, I have a question. So, like for Adora, at 3 years, in this ESMO, the curves were, when you stop osimertinib, the curves were starting to dip earlier again. So can an MRD or a ctDNA analysis be done at that time, that people who probably have it positive or negative, and then whether to continue it for 5 years, 6 years, or stop at 2 years, 3 years?
I don't want to, I can tell you what my guess is. I could be wrong, very wrong. And I don't know this for a fact. So I want to preface that very carefully. But in reality, what happens in these tumors, minimally, there is no, as I told you before, your ctDNA is very unlikely to be positive in these situations after 2 or 3 years in many instances. What happens is that there are clones that are in the resting phase and they are dormant somewhere. Once you stop these things, they start proliferating. And in fact, the interesting would be not the ctDNA when you 2 years or 3 years later, but after you stop, after 3 months or 6 months, you will see them increasing before there's a clinical disease. So that's the group that would tell you ahead of time, these are the people who are going to relapse. But I suspect that many of them would be negative at the time you stop therapy because there'll be very minimal burden disease. But it really goes back to the basic biology. I think the TKIs, I've always been very skeptical of this. I don't want to take away the focus of the conference. The true endpoint is overall survival. That's why, despite a lot of pressures, even from FDA, I kept the overall endpoint for the Alchemist trial to overall survival and not disease-free survival. Disease-free survival in early stage lung cancer adjuvant therapy, in my opinion, is not the right endpoint. It's overall survival. You are going to give a finite therapy, you're going to increase the cure rate in this patient. That's what matters. If you merely delay the recurrence or relapse, at the end of the day, these patients do just as well, and the overall survival is the same. Whether you do the after surgery or at the time of relapse, it's a pointless exercise. That's why we are getting ourselves into this useless, into this knot. I also think that the TKI inhibitors the problem is that they don't eradicate disease. We have seen that repeatedly in metastatic disease. They are unlikely to improve the overall survival. And you will see this relapse. You've seen this before with the smaller Tarceva studies that we did earlier on. As you stop, the thing, this is what expected, is what many of us expected, not a surprise. So I don't think they are the means to cure these patients. Eventually, we have to understand what else is driving these tumors, and that's the way to cure these patients. We are not there yet. That may be a combination of some regimen, and we still don't understand what happens to these tumors after they get transformed by EGFR mutation. So that's where things come.
Thank you, sir. So continuing to learn from our experts and shifting gears, and we'll just discuss quickly 3 case scenarios. And so I request Dr. Govindan and Dr. Ullas Batra to participate in the discussion. So the first case here, we have a 55-year-old a non-smoking man diagnosed with stage 4 adenocarcinoma lung with multiple bone metastasis. The tissue was limited, just like Dr. Ullas mentioned. Tissue is sometimes an issue. EGFR, ALK, ROS1 testing for common alteration done, negative test. The PD-L1 expression was more than 50%. So, Dr. Ullas, this question is for you because you spoke about the liquid biopsy. So there was limited tissue with which they did the hotspot mutations for this EGFR, ALK, and ROS1. So the question is, if these 3 are negative, would you go ahead and do a liquid biopsy at this stage?
I'll do a repeat tissue biopsy.
Repeat tissue biopsy. Okay. And what is the role of liquid biopsy at this stage?
So you can send that if the patient really refuses, as I told, liquid biopsies, the sensitivity is only 80%. And it's not very good for the fusions and stuff like that. So probably I would counsel the patient for a for a tissue biopsy. I am very skeptical of starting a patient on immunotherapy before a tissue biopsy and a small panel NG is done on this. But if the patient absolutely refuses, I'll go ahead with a liquid biopsy.
Perfect. So this patient was started on carboplatin with pemetrexed and pembrolizumab, and the patient progressed after 6 cycles. So again, Dr. Ullas brought out that very nice point of doing a ctDNA even with in patients on immunotherapy. So after 6 cycles of treatment, the new PET-CT shows progression. I mean, I would say this could be even IUPD, increased disease size of primary lung lesion, new bone lesion, liver lesion, and mediastinal lymph nodes. Dr. Govindan, in fact, I have troubled you with these questions previously on telephonically. So the question is, when would you consider this as a pseudoprogression or IUPD, and when would you consider changing treatment, say at the end of 6 cycles, like this patient?
What is IUPD?
Immunotherapy unconfirmed progressive disease based on the iRECIST 1.1, where the pseudoprogression.
You know, a pseudoprogression is more imagined than real. I have not seen a convincing pseudoprogression in lung cancer ever in my life. And so I think it should be important to keep in mind. It happens in melanoma to some extent. So most progressions are real. I don't think there's pseudoprogression in general. At least I've not seen, and it's not been very convincingly reported in my view in lung cancer. And there's been some reports in the early stage lung cancer where the PET is more active from tumor necrosis, and that's a different situation. When you have multiple new lesions, it's honestly unlikely to be pseudoprogression. I think I agree with Ullas that I think looks like this patient could have some accessible areas for biopsy. If it is, getting that is important in all patients, important particularly non-smokers, because upwards of 60% have potentially targetable alterations with routinely available drugs, and maybe up to 80% if you include the clinical trial that was the subject of our paper in JCO last year. So it's really important not to miss that, and I would go after that. In the absence of that, liquid biopsy is not unreasonable, particularly when you have so much of bone disease that chances of false negative are probably lower. But still, tissue is the best thing if you can get. I agree with Ullas completely. I'm sorry, the question you asked is what?
The question I asked is, would you consider pseudoprogression at this stage and continue with the immunotherapy, or would you consider changing treatment?
No, this is real progression.
Sure. Dr. Ullas, would you correlate with the ctDNA? You're doing a study on nivolumab and ctDNA.
I'm glad you asked that. That was again a very research study. It has taken 3 years for us to do that study. We don't do it in clinical practice at all.
Sure.
We don't actually. I would either get a tissue biopsy done and get an NGS done on this patient, or again, if the patient is not willing, get a liquid biopsy done.
So just like the last point you mentioned, Dr. Ullas, at progression, since the patient was not willing for a tissue biopsy, the liquid biopsy was done. And they found these two mutations, the S768I and the G719A. So what do you think? What would you do with this? And not the canonical L858R mutation or the exon 19 mutation. What would you do in this kind of situation?
So I'll go back to my initial EGFR report and then see what was the tumor celerity in that. Was it a true negative report which came? I would also look at the, whether there was a good tumor content in that, whether the lab was good, whether it was sanitized or not. I would probably not send the sample again to that lab, not just joking, but I will give afatinib. I'll give a TKIs to these patients after that. Afatinib, I think, is what I would give.
So so that's a very fair point, Dr. Ullas. So I just want to know between, just for the interest of our fellows, between the different TKIs, between erlotinib, gefitinib, afatinib, and osimertinib, why did you choose afatinib in this kind of situation?
The data is more stronger at a combination of Lux lung trials. I think 3, 6, and everything is more common with the afatinib in rare mutation. And there was some retrospective study that gefitinib or erlotinib are not that active. There's some small data of around 50-60 patients with osimertinib also, but the drug approval is there for afatinib, and that's what I would do.
Sometimes we see these uncommon mutations in combination with the L858R. In that case, a common with uncommon, a combination, a compound mutation. How would you address that?
Again, afatinib would be my choice. However, again, depends upon the patient. If it's an elderly patient, I would not even desist from giving osimertinib in those people also. But if the patient is young, fit, afatinib is reasonably okay.
Dr. Govindan, any difference in thought in this situation?
No, I agree with Ullas. In fact, interesting you asked this question. I had a fellow in my clinic yesterday, and she saw 2 patients on afatinib, and she was quite puzzled because the first-year fellow never heard of afatinib, and I was explaining to her one of them a similar instance, and both of them have been on afatinib for 7 years, never progressed. And again, I'm just taking it off, it's an anecdotal example, and I'm not promoting uncommon mutations. I agree like this, and it's I totally agree with that approach. The only thing I would do is the 40 milligram is probably a wrong dose for afatinib for most patients. I don't know what your experience in Indian population is. 30 milligram is best tolerated in my view. So I almost always start them on 30 milligram. I have a handful of patients on afatinib. I began a while ago, and they're all on 30 milligrams.
I would agree one point. I start them on 30, keep them on 30. I don't go to 40.
I agree. Me too. I don't even start at 40 anymore. 30, yeah, I agree.
Great. So this was the complete exon sequencing versus, so that's how it got picked up. And so that you don't miss these uncommon mutations. And we know that EGFR, even these uncommon mutations, certain times it could be predictive of a poor response to IO. So it's very important we pick up these mutations. So the patient was offered afatinib. The patient achieved a partial response after 3 months of treatment. That is how much we have follow-up from this particular case. Dr. Santosh, would you like to take this case?
Yes. This is a 59-year-old non-smoking woman diagnosed with stage 4 lung cancer, adenocarcinoma with multiple visceral metastasis. A tissue NGS was done, and a L858R mutation was identified, and she was started on gefitinib. Patient on the TKI progressed after 10 months of therapy. At progression, a liquid biopsy and an NGS was performed, as the tissue biopsy that was attempted was inconclusive. And the Guardian 360 test found that the patient had an EGFR L858R mutation and a RB1 mutation and a AR and a EGFR T79M mutation. And therapy with osimertinib was offered to this patient. At this point, I would like to ask Dr. Govindan, if such a report comes to you, how would you like to approach this case? How would you comment on these mutations that has been picked up on liquid biopsy?
The only thing is the T790M was not present before.
It was not present before, yes.
Okay, I think osimertinib is a reasonable choice. Sorry, I had to answer my nurse's question. I had to take care of some things. So was this person on gefitinib or erlotinib before?
The patient was on gefitinib for 10 months and progressed
Okay.
on gefitinib.
Okay, I think osimertinib is a reasonable choice. The only thing I would caution is that whenever there's an RB loss in a sample, I get a little concerned, and especially the rapid progression when they have they are the people with high risk for transformation to small cell. And again, usually you see both RB and P53, and very often you see, or at presentation they have P53 mutation and NF8 or something, and then at progression we begin to see RB1, and then in a month, eventually they become small cells. So you have to keep an eye on that a little bit. That's where the biopsy of the tumor tissue is helpful to make sure that the histology hasn't changed. But in a person like this, probably it is still adenocarcinoma. So I would give osimertinib. I have no concerns. I wouldn't be distracted by the other alterations. It's the T790M is what's the important thing.
Okay, so despite the RB1 mutation, you would still, if there's a suspicion of a small cell lung cancer, would you rebiopsy to look for a small cell lung cancer?
If the presentation is concerning, like a rapid progression, lots of significant disease in the liver suddenly, then I would biopsy, sure. If there is a kind of a slow indolent progression, RB1 alone, I don't want to misrepresent facts here. RB1 alone doesn't tell you that it is small cell. RB1 loss is reported repeatedly in lung adenocarcinoma and even some squamous. It's the loss of P53 and RB1 together that is hallmark of small cell cancer, and we see that in transformation. Not only lung adenocarcinoma, but also in prostate cancer when it becomes neuroendocrine differentiation. It's something that transcriptionally that changes it. And so I wouldn't be distracted by that at all. It's a T790M that matters here.
Govindan, what's your take on this combination therapy of chemo plus TK? It is very attractive to Indian physicians, where you can give a combination and increase the PFS in the first line, the combination of chemo plus gefitinib.
Yeah, it's an age-old question. When we combine these drugs, you have a little better PFS, but if you do sequential therapy, you may end up doing that same, or eventually, overall survival would not be affected. So at least in the US, and I definitely don't do combination therapies with chemotherapy. I'm well aware of the TADA presentation. But I think there is an ongoing study with osimertinib with this combination chemotherapy. I'm very skeptical, and I think one of the advantages of being on osimertinib for patients is a near normal life. They don't have to come repeatedly; they come once in 2-3 months. I don't see that addition of chemotherapy significantly changes. You definitely will see an increase in PFS, but if you look at overall survival, it does not change. The same with the Avastin or a bevacizumab combination of VEGF inhibitors with the EGFR inhibitor. You see increase in PFS, but there's no real increase in OS. And the Japanese data, we try to do a study in the US. We did that study, I should say. And again, the results are not that impressive. So we have not adapted to that. So we are not, in fact, I don't know if very many US physicians, academic or community, combine chemotherapy with osimertinib or gefitinib. I remain unpersuaded that the combination is any better than giving chemotherapy at the time of disease progression. I would save it for that at that point.
Okay. Thank you. So this patient received osimertinib and did progress after 3 months, and a repeat biopsy did show a small cell transformation.
Oh, I'm sorry, I didn't. Oh, that's interesting.
So for want of time, we'll move on to the next one. I'll leave Dr. Vishwanath to take this forward.
So, Dr. Govindan, just one last question about this particular case. Now, this patient has got transformed to small cell lung cancer. So, between atezo, durva versus plus chemo, what would you do in this kind of situation? Transformation to small cell.
Small cell cancer is cancer. You would think, if you didn't know anything and if you were living under a rock, you would really think that small cell would be the one who would be cured with immunotherapy. Why? Because all of them are smokers, 95%. The tumor mutation burden is far to the right, almost even right to the melanoma. And you would think with so many, you would think they'll respond well to this. But in reality, the response to immune checkpoint inhibitors is pretty unimpressive. There's a marginal improvement when you add chemotherapy to immunotherapy. It all is context. Would I sell a farm and a house and destroy a family to give durvalumab or tocilizumab in small cell in a resource-constrained place? Absolutely not. I would argue even in a resourceful place like this, we have to think twice about it. Sadly, in oncology, we are trying to gloat over a 2-month improvement in overall survival or with this. In reality, it doesn't really make a big difference. They're enormously expensive, so I'm not that persuaded. If I were practicing in India, I would just give carbetociclib. In the US, for various reasons, we end up giving it, but I'm completely unpersuaded that the immunotherapy does anything for small cell at the moment, although granted the 2 studies have shown a modest improvement in overall survival. But we have to really understand the mechanism of resistance. And as I said before, we did discover immune signaling pathway activation in small cell that may contribute to resistance. So we have a long way to go. But I would definitely think twice before using these expensive drugs in these patients. Like, again, cost is important, but more the science also is questionable here, in my opinion. I would give carbetociclib. You will always see EGFR mutation in these tumors, and you shouldn't get distracted because the EGFR mutation is present at the birth of the cancer cell. So called founder mutation will be present in all subsequent ones. So that has no bearing on the disease. It's just gone a different direction now.
Dr. Ullas, you have a question.
Yeah, so I agree with Dr. Govindan in being very skeptical about Adezo and Durva. What is your take about the extrapulmonary small cell carcinomas and use of immunotherapy in those? So you have the maxillary sinus and esophageal. A lot of time people ask us this question. Dr.
Yeah, yeah, Ullas, I don't have a personal experience, but I know the biology a little bit, and they behave almost similarly. In fact, even the prostate uroendocrine behaves in a similar fashion. They've not been adequately studied, in my opinion. I would probably stick with carbetociclib.
Thank you. Let's quickly go for the last case, just a couple of slides and we can wrap it up. 85-year-old non-smoking woman diagnosed with stage 4 adenocarcinoma lung. Tissue testing identified the canonical EGFR L858R mutation. Patient was treated with osimertinib. And then the patient's disease progressed. And I think, so Dr. Ullas elucidated this very well, the resistant mechanisms. So the patient had EGFR L792H, EGFR C797S, and the EGFR L858R. So Dr. Ullas, your thoughts on this?
I was hoping you don't ask me this question.
Last one is also quite interesting.
So MSIH in lung cancer is less than 1 to 2% of patients. We don't regularly test for MSIH. MSIH on liquid biopsies is even more. I don't even know the technology that they have used. If you have a tissue biopsy available, I would rather do the MSI testing and the MMR testing on that would be skeptical in giving immunotherapy to a patient of lung cancer with MSIH. Very skeptical before I have more other things. Not sure about the validity of this test. Dr. Govindan.
I completely agree with Ullas. Every bit, every word. I completely agree with him. Nothing to add.
Dr. Ullas, what do you have to say about this? If you have a loss of T790M on osimertinib, or if you have a cis mutation with the presence of T790M, or
I think that's very easy. If your T790M is present, look at the mutation in cis or in trans. If you have it in cis, go to 4th generation. If you have in trans, go to 1st plus 3rd generation combination.
So what is your experience on this?
Very few. I mean, 2 patients who had got this, nothing, no great experience. So one, it's like a single cell study. Maybe Dr. Govindan will have experience.
Not really. I'm very aware of this, but unfortunately, I think it's a little. I honestly think we need better therapies for this, and I would go with chemotherapy instead of wasting time on this.
So we've also seen this kind of a scenario in several other malignancies, like including colorectal. You give EGFR blockers like cetuximab or panitumumab, and then you go to Bev, and then they say that there could be sensitization. The patients might have sensitivity to this EGFR blockers again because of the resistance clones going down. So is this an area of intense research, Dr. Govindan? Like when you give a break and then probably
Yeah, maybe. And I think eventually, once you give up, you cannot give intermittent therapies that can promote resistance too. So you have to give it for reasonable time. Yeah, I think what happens is that at the time of initial transformation, you need some gene alterations like KRAS mutations. If you do that alone in cells, you only, in vivo experimental genetic manipulation, you can only get adenomas, not adenocarcinomas. For them to become carcinomas, you have to lose tumor suppressors, and they activate certain pathways and programs we understand not very well in many instances. So that what determines the course of a disease. EGFR mutation is important for the initiation, but not sufficient to cause cancer. The other things are important. And that I think we understand very poorly. When we can understand first better, and second, we are able to manipulate this, then we'll begin to see some cure in those populations. And that may come either through informed biology approach, or eventually may come through random efforts like a combination of some immunotherapy, chemotherapy, and that kind of blunt instruments that we have that doesn't really need a specific mutational alteration. So I think it is, and I think that's the kind of an approach you can use.
Anyway, this patient underwent treatment with pembrolizumab and chemotherapy based on the MSI high. In fact, that's why the patient received pembro, I suppose, and then CR was achieved after 6 months of treatment. So that wraps up our webinar. Very interesting webinar. Thank you so much, Dr. Govindan and Dr. Ullas, for those wonderful talks. Any last thoughts, Dr. Govindan? One thought from each of you.
No, I think I really enjoyed every minute of it, even though it was a little, it's not, I've been going through a lot of things lately, but I enjoy every second of it, and I truly, truly appreciate the chance to participate in this. Great job.
Thank you. Dr. Ullas, your final thoughts.
I think research is something which is very dear to me. And I think as young oncologists, and not so young me now, we should invest in research. That is the only way we can go up to the standards that Dr. Govindan spoke about. If you have a video of this, please do share with all the younger colleagues. If we can inspire the next generation, that's the way to catch up with the rest of the one. We are making a small step, but we need to make a lot of strides, and research has to be priority in terms of medical oncology.
Yeah, very well said, Dr. Ullas. Thank you, both the speakers, and thank you our sponsors, Garden 360, for the support for the educational grant, and all the attendees who have logged in from various parts of the country and abroad. And of course, our fellows who have also joined in large numbers. I'm sure they would have DNB and DM residents who have definitely benefited from this. I'm sure they would have benefited. Thank you so much, and looking forward to have many more webinars with both of you.
Thank you.